Hi E12s, welcome back. We're going to talk about CRISPR-Cas9 in this video and a few GMOs that can be created from it. All right, so what is CRISPR? All right, so CRISPR stands for obviously something. It is an acronym, so it stands for Clustered Regularly Interspaced Short Palindromic Repeats. Okay, so in other words, they're clustered, they're every now and then going to be spaced, they're short, and they're palindromes, and they're repeated. Okay. So the repeats are interrupted by what we call spacer DNA. So you can see that here. All right, there's your spacer, which really is the cell's machinery. Okay. So obviously we're looking at bacterial cells here because this is what they were found to have as a defense mechanism. So it's a viral mugshot. So looking at viruses, and the spacer DNA essentially is their specific defense to those viruses. Okay, yellow is not a good color. Green, so there's your spacer DNA. Okay. So CRISPR sequences are always downstream of the gene for Cas9. So if this is a DNA sequence, all right, the Cas9 gene obviously is going to be transcribed and translated to produce the Cas9 protein, and that happens before the CRISPR array. All right, remembering this is just a prokaryote, it's like some sort of bacterium's genome. So what is CRISPR? It's a naturally occurring adaptive immune response that's done by prokaryotic organisms. So if you look at the E. coli here, all right, it's getting attacked by these bacteriophages, which are viruses. Okay, they're just a couple of proteins and some nucleic acid. So when a bacterium encounters a virus, in this case the bacteriophage, all right, it takes a mugshot or a memory of its nucleic acid. So it could be DNA, it could be RNA, all right, by storing some of the viral genetic material with its own genome. So essentially it takes the injected DNA, because if we just think back to what viruses do, they inject their own DNA and they hope that this DNA makes its way into the genome of the organism. All right, so therefore it's now in here, and the proteins to create more viruses are then going to be expressed. Okay, that's what they aim to do. But in this case, they have restriction endonucleases, certain bacteria that will cut the nucleic acid that's injected at a certain site. We call that the PAM site. Okay. So how bacteria use CRISPR? So essentially, all right, you have your bacteriophage, as you see here. It's injecting its DNA. Okay. So we have the Cas1 and Cas2 proteins. They work first. All right. So Cas1, Cas2. They do the job first. And they essentially will cut a specific part, all right, of a brand new viral piece of DNA. Okay, so if this is a new virus, they'll cut that, Cas1 and 2. If it's an old virus that's already injected its DNA, then Cas9 will be cutting that and destroying it. Okay, so keep that in mind. So what Cas1 and Cas2 do is, if they encounter a new virus, and therefore new DNA, they're going to take a part of that out. Okay, and they're going to find a particular PAM sequence. All right, protospacer adjacent motif. That's what PAM stands for. They're going to find that in this piece of DNA. Okay, and they're going to cut that out on either end, and they're going to take that and put it literally into the CRISPR array, which is the bacterium's own genome. So it's going to add it to its own spaghetti DNA, that I like to call it. All right, from there, essentially CRISPR-Cas9 can use that, all right, through various different methods. It can use that to cut viral DNA when it comes in. All right, and when it cuts the viral DNA, obviously that's going to break it in half, and then it's unexpressible. Okay. So yeah, this is a pretty fascinating process that they do. All right, we're going to look at it in more detail in the next video as well. So you have the protospacer that gets turned into a spacer. As you can see, this one's yellow, but could also be a different color, representing a different virus. All right, so this might be virus X, this might be virus Y, and this new yellow one is virus Z. Okay, something along the lines of that. Alrighty, so what's the application? Essentially, what do scientists do in the lab? So really, CRISPR-Cas9 can be harnessed by scientists to cut a particular thing. And this is the Cas9 enzyme here, in the background, and that attaches and it locks into a particular DNA sequence. Okay. Now, generally, that's the spacer sequence that we spoke about, which is going to be, expressed in a different way, and then created into sgRNA, okay, which will then go into and lock in to the Cas9 enzyme. All right, and remember the Cas9 enzyme is an endonuclease, so it's going to find that exact matching sequence, and it's just going to cut there, all right, like all endonucleases do. In most cases, it's going to cut a couple bases down from this PAM sequence. All right, so it might cut straight through here, which is 4 or 5 bases down from the PAM, which you see here. So that's that NGG, all right, which represents a nucleotide, guanine. So it can be anything. It could be UGG. Oh, actually, sorry, it could be AGG, TGG, CGG, or GGG. All right. Yeah. And really, so what that means is that cuts a specific piece of DNA. All right. So the application here really is that if that cuts a specific piece of DNA to defend the bacterium from a virus, can researchers and scientists harness that and insert their own piece of DNA that's complementary to a gene that we want to cut? All right. Why would you want to cut that? We'll have a look in the next slide. All right. So scientists can then use a eukaryotic cell's own repair mechanisms to alter the DNA. Okay. So essentially, scientists can cut at a particular part using a Cas9 enzyme, and if it's a eukaryote, all right, it's going to try and repair that DNA. But often it's a process called non-homologous end joining, among another one. That really is error prone, and that breaks very easily. Okay, so in other words, we can cut a piece of DNA. It's going to try to repair, but it won't repair properly. Okay, and therefore it's now a knocked out gene. All right. You can see that in the process here. So target sequence of DNA, you cut that out and you load it up into guide RNA, which is this shape here. Okay, that shape then locks into a Cas9 enzyme. It like hooks in. All right, and that's now its guide, because guide RNA guides the enzyme to where it needs to cut. The enzyme will open up the DNA, as you can see, and then it will cut a couple bases down from the PAM sequence. All right. Afterwards, the cell will try to repair it, but it will be error prone. It will be done very dodgy. It's like getting a really dodgy tradie to come in. They'll do the job, but it'll break down. It might not work properly, etc. All right. So really, how can we use CRISPR-Cas9 to make genetically modified organisms? So that's what a GMO is. GMO, organism that has had their DNA altered using genetic technology. Okay, so it can be any genetic technology, but in this case it's CRISPR-Cas9. Yeah, we can use it to do a bunch of different things. Okay, firstly, you should understand that we call these a transgenic organism if they've had a gene inserted from another species into their genome. All right, and once that's done, then it's become a transgenic organism. And that gene we call the transgene. All right, gene being transferred between species. All right, so what are some examples? We can make celiac-friendly wheat, so wheat that celiac people can eat, which is a net positive gain. potatoes that don't release acrylamides, and other stuff when you fry them, for example, which is a carcinogen, so that leads to cancer. High fiber bread, as well as the golden rice that we should be aware of, which is high in vitamin A and is grown in second and third world countries to reduce the rates of blindness in children who have vitamin A deficiency. All right, these are really the uses for CRISPR-Cas9. We're going to talk about ethics in the next video. So this is just more of an introduction to CRISPR. Okay, so thanks for watching. Have a nice day.